Without further ado, welcome back, everyone. Thank you for joining us again. This time, I would like to introduce our keynote speaker, Dr. Chris Barty. Dr. Barty, he received his bachelor degree in chemistry, physics, and chemical engineering from North Carolina State University, then went on to receive his PhD in applied physics from Stanford University. His professional appointments have been all up and down the coast of California, from Stanford to UC San Diego, and then to Lawrence Livermore National Laboratory in Pleasanton, California, where he was most notably the Chief Technology Officer of both the National Ignition Facility and the Photon Science Directorate. In 2017, he was hired as a distinguished professor with a primary appointment in the Department of Physics and Astronomy at UC Irvine, where he also serves as the senior faculty member of UCI's newly minted Convergence Optical Sciences Initiative, or COSI, which is a collaboration between the Beckman Laser Institute and Beale Applied Innovation here at UCI. Dr. Barty is also the co-founder of Lumitron Technologies, whose headquarters are located in the UC Irvine Research Park, just across the street from the medical education campus. If you have any questions during the presentation, feel free to ask them at the end of the presentation or ask them in the Zoom chat during the talk. After the address is concluded, I will moderate questions from the audience from both raised hands and text questions. And to raise your hands, please click the reactions button at the bottom of the Zoom window, click raise hand. So I'll do my best to get as many questions as possible in the time we have allotted. And without further ado, please give a warm virtual welcome to Dr. Chris Barty.
I feel virtually welcomed. Thanks very much, Hatham. So thanks everybody for joining in this afternoon. I want to tell you a bit about why I'm here in Irvine and how it came to be that I'm here in Irvine and the background behind this technology that we think is going to be a revolution in clinical X-ray systems and applications, and in other applications as well, as you'll see in the talk. And so to kind of give the bottom line up front here, if you think about the sources that we're making now, that we're going to be making in our labs and in the Irvine area, they're all based on a technique called laser Compton scattering. Laser Compton scattering, when it transitions out of the lab and into the clinic, will be the first real change in clinical or commercial X-ray technology in over 120 years. There are other kinds of X-rays that are used in a science vein, but not in the clinic. And so this will be an interesting time. There are a number of unique attributes to the laser Compton sources. In particular, they have a lot in common with respect to their output in terms of billion-dollar scale science facilities. I'll explain why. But they are very compact. I would say large desktop scale. If you're talking about clinical X-ray energies in the kind of 50 to 60 kilovolt range. And that means that they occupy a footprint that's nominally the same as a modern CT or a modern MRI. From a performance perspective, they allow you to image with X-rays at up to 1,000 times higher resolution than you currently do in clinical environments. They also allow you a number of different avenues for reducing the dose to the patient in an X-ray exposure. And some of those avenues are quite dramatic, up to 100x. And then it also, because it's a tunable monoenergetic source, there are ways to use the monoenergetic nature of the photons to determine the composition of the objects that you're looking at. In particular, to tell what isotope or what element is contained in that object. The company side of things is very aligned with other medtech activities, and the consultants with it are leading physicians around the world, one of which is Dr. Phil Stricker, who's the director of the Australian Prostate Cancer Research Center. His view of this technology, and of course I share the same view, is that if we do what we're trying to do and it works the way that we say that it's going to work, you may never remove a breast or a prostate again. You may be able to discover the disease, treat the disease before it gets to the place where you have to be so dramatic. So let's talk about a high-level technology comparison. If you think about clinical X-rays today, the way that the X-rays are made is really the same way that Wilhelm Roentgen discovered X-rays in 1895. You take an energetic electron beam, you slam it into a metal, and as that electron goes into the metal, it bends its path. And every time you bend a path of an electron, you're accelerating the electron, and every time you accelerate an electron, you radiate. And the result of that stopping process of the electrons is a broad spectrum of X-rays that extends at its upper end to the endpoint energy of the electron that you used, and at its lower end to zero. And it peaks somewhere in the middle. And it spits those X-rays out into all angles. So if you're looking at this kind of source, I think the right analogy is a flashlight. Produces white light, and it produces a wide beam of things going out in all angles. Laser Compton source isn't a coherent source, but it shares a lot of properties of a laser. It has a very collimated beam, a beam that's divergence is similar to the divergence of a laser pointer. It has the ability to produce on axis a very narrow, very pure color of X-ray, and that bandwidth of those X-rays is on the order of the bandwidth that you see in, say, a green laser pointer. And of course, we can tune this source by changing some of the parameters in the system, so we can tune anywhere in the X-ray spectrum that we want. The technology that is here in Irvine now is actually based on my group's work at Lawrence Livermore National Lab over a span of about 17 years. There were 3 generations of machines, laser Compton machines, that were built at Livermore for different applications. Those applications were basically around nuclear security, because Lawrence Livermore Lab is a nuclear weapons lab, a nuclear weapons science lab. The 17 years of activities included about $220 million worth of R&D between the efforts at Livermore and the efforts at the SLAC National Accelerator Lab over at Stanford University. So Compton scattering, how does Compton scattering work? The way that it works is you take an electron beam and you accelerate it to near the speed of light, and then you do a head-on collision with an electron beam, with a laser beam, with that electron. That laser beam has a transverse electric field that is oscillating. That transverse electric field causes the electron to have a transverse motion. That transverse motion is a radiating dipole. And because the electron is moving at nearly the speed of light, what you see in the laboratory is the Doppler upshifted output of this radiating dipole. And that Doppler upshift can be very big. It can be 10,000 to 1,000,000x what the laser photon is that comes in. And by changing the energy of the electron beam, we can change the color of the laser photons that come out, or of course we could change the color of the laser photons that are being used, and that would also change the color of the X-rays that come out. Okay. Now, all of that sounds great, but there's a big problem, and that big problem is that the probability of an electron interacting with a laser photon is very small in this Compton scattering process. That probability has a cross-section of about 6 times 10 to the minus 25 square centimeters. That's a very, very small probability. And so the only way to really get an appreciable amount of photons out of this process is to put all of the electrons and all of the laser photons into a very small volume in a very short period of time. Compton scattering was first demonstrated in 1965 with a giant ruby laser, and the first experiment made 8 photons. We're trying to make sources now that have of the order 10 to the 13 per second. So it's a very different arrangement. To get all of the photons in the same place in the same time, what we do is we start with a common laser. We make a train of laser pulses. We split that into 2 trains. One train is converted to the UV and hits a cathode to make electrons. The other train is amplified, converted to green light, and collides in a head-on way with the electrons to produce our yellow X-rays. Everybody knew that X-rays were yellow, right? Yeah, you know that. This particular geometry is a geometry that, if done correctly, allows close to unity efficiency for the conversion of electrons into X-rays. That is, every electron would scatter and produce an X-ray. It also allows you to make a very pure color for the on-axis photons, which is important for a number of applications, and actually was a driving force behind some of the Livermore activities. The other thing that's interesting is you make a very small scale spot size when you do this kind of arrangement. You can make anything from 10 kilovolts to 20 megavolts if you really want to. It just depends on the energy of the electron beam that you have in the accelerator. Okay. If we think about the comparison of the technologies, clinical and industrial X-ray sources are really all based on the same Roentgen tube that was from the 1890s. Roentgen actually accidentally discovered X-rays. He was firing an electron beam and doing some studies and was trying to take images, photos of the electrons, and he put a block in front of the film by accident, but then found that the film was still exposed. And so something had gone through the block in front of the source and exposed the film. And after some looking around, he discovered that, well, there was a new kind of radiation that was coming out of this interaction of the electron beam with a metal, and those were X-rays. Very quickly after that, 1896, I think you have a picture of Roentgen taking a picture of his wife's hand with the ring on the finger. It's a very famous picture. I think he probably gave his wife a little too much dose in that picture, but hey. For this discovery in 1895, he was the first awardee of the Physics Nobel Prize. So it's long history. What we do in a Compton source is really much different than what happens in a Bremsstrahlung source. It has much more to do with what happens in large-scale international X-ray science facilities, which are called synchrotrons. Synchrotrons are giant circular accelerators that have nominally a kilometer footprint, and periodically around them they'll have an array of magnets that are used to cause perturbations in the electron beam. What we're trying to do is take this billion-dollar scale kilometer footprint and get it to the million-dollar scale with a meter footprint. If you think about a synchrotron, the way it works is you have an electron source, an electron accelerator. You create a bunch of electrons. That's a short high peak current arrangement of electrons, maybe 10 to the 8, 10 to the 10 electrons. They have an energy that is above a GeV, 1,000 MeV in this case. And then you pass it through a periodic array of magnets that have their poles alternating. And that periodic array causes a transverse kick on the electron beam. And so the electron beam creates this wiggle. That wiggle is a radiating dipole. And what you see in the lab, again, is this Doppler upshifted light. And the spacing of the magnets in this arrangement is on the order of a centimeter. In a laser wiggler, what Compton source is sometimes described as, you have not GeV electrons, but maybe 100 MeV electrons. But now the spacing that you have is the wavelength of the light that you're using, and that can be submicron. That 10 to the 4 difference, that 10 to the 4 difference in spacing is the thing that allows you to make the sources much more compact. You kind of gain a factor of 10 in the energy, and you lose a factor of 10 to the 4 in the spacing, and you kind of end up with 10 to the 3 in compactness if you have a laser Compton source. So fundamentally, that's how we get to a source that might look like the size of a modern CT machine. If you're going to compare X-ray sources, one of the metrics that people use is the average brilliance of the source. The average brilliance is the number of photons that come out of a given aperture into a given solid angle with a given bandwidth. And when synchrotrons are built, they're built with these curves like the blue light here that tell you what the brilliance is that's coming out of the source. Synchrotrons, by and large, are used as X-ray diffraction instruments. They're used to, in this kind of 10 kilovolt to 20 kilovolt range, where the wavelength of the X-ray is roughly the spacing of atoms and molecules. And if you put a coherent beam or a quasi-coherent X-ray beam onto a crystal, a protein crystal, let's say, the diffraction pattern you get will allow you to work backwards and tell you what the crystal was and what the structure was that you were looking at. So the vast majority of work on these devices is for doing this kind of crystallography. They tend to die out at around 100 kilovolts, and they get really low in output when you get above 100 kilovolts. They die faster than exponentially, but that's because they were never designed to go out to this range. You would have to make a much bigger loop and a much bigger energy to move out into the megavolt range. Rotating anodes, if you put them on this plot, sit somewhere down here in the 10 to the 7 kind of range on average brilliance. Compact laser Compton sources follow this red line trend. Compact laser Compton sources actually get better the higher the energy you go, because the problem with a Compton source is getting the electrons into a small spot. The higher the energy you make the electron, the smaller the spot you can focus it to, the electrons become stiffer. You have to have stronger magnets to focus it, but you can get to a smaller spot. And that smaller spot allows you to get square more photons because it's a cross-section activity. So you're getting square more photons out of a square smaller area. If you're looking at brilliance, you have photons in area in the opposite end. The brightness is going up something like the 4th power of the electron beam energy as you move up. If you look comparing to a synchrotron, sources, compact Compton sources are a few orders of magnitude below a synchrotron in the 30 kilovolt range. But compared to a rotating anode, they're 10 million times brighter, right? So it's not that we're competing directly with synchrotrons, it's that we're really doing something very different than what you have in the clinic today. If you go out to a couple of megavolts, Compton sources are unique. They're 15 orders of magnitude brighter than the best synchrotron on the planet. And then it becomes quite an interesting story. Above 100 kilovolts, the photon no longer primarily interacts with the electrons around the nucleus. It can now interact with the protons that are in the nucleus, and it can cause resonant motion of the protons in the nucleus. And there are all sorts of nuclear things that happen. In fact, the origin of most of the funding at Livermore was a white paper that I wrote back in 2004 that said, you're going to be able to do nuclear science, nuclear applications with photons for the first time if you have these kinds of sources. You could make pulse positron sources, you could make isomer physics, you can do photofission if you make high enough energy photons. And if you can do photofission, you can make micron scale picosecond duration neutron sources, and there are all sorts of applications of those sources. Many of those are summarized in this paper around the technology. During the activities at Livermore, I coined a term called nuclear photonics to be the representation of all of this field where you can use tunable monoenergetic photons. The first International Nuclear Photonics meeting was held in 2016 in Monterey. It's now being held every other year. Well, it was supposed to be held last year in Kirishiki, Japan, but COVID took care of that, and now it's being held virtually in Japan this June. But it's now become an international conference series, and there's a wide community of people who are pursuing nuclear photonics and designing and building machines for this kind of application. One of the things that motivated the activity at Livermore was nuclear resonant fluorescence. Nuclear resonant fluorescence is an incident gamma ray hitting the nucleus, causing a resonant motion of the protons in the nucleus, and then relaxing and releasing a photon as an output. That is a direct analogy to what happens when a photon comes to, a visible photon comes to an atom or a molecule, and it's resonant with the atom or molecule. If I had a 589 nanometer yellow photon and I had a sodium atom, I would excite the outer s electron to the first p orbital outside of it, and I would absorb that 589 nanometer light very readily, a big cross section for that absorption, and that atom would relax by emitting the 589 nanometer light into random directions. The same thing happens with the nucleus when you have tunable gamma rays. This is a spectrum of strontium-88. You can see there are many, many lines that are here. These lines depend on the number of protons in the nucleus and the number of neutrons in the nucleus. So they are signatures of the isotope, not of the element. And that was very important because that allowed us to do isotope-specific material detection. Most of the resonances occur in the kind of 1 to 3 megavolt range. That's a very interesting range because it's a range where the Compton scattering cross-section for most materials that causes absorption is decreasing, and the pair production cross-section hasn't increased. And this region is generally known as the universal transparency window. Every material has transparency in this kind of 2 megavolt range, which means that you can look through very thick objects and see something on the inside. One of the first things that I suggested at Livermore that you could do was to try and find uranium-235. For those of you who aren't nuclear weapons experts, there are 2 things that you can make a fission bomb out of, plutonium-239 and uranium-235. You should never worry about somebody trying to smuggle a plutonium-239 weapon or even a dirty bomb into the country, because both of those radiate tremendously, and we have plenty of very sensitive radiation detectors that will sense the presence of those objects. Now, you could argue that somebody could shield it with enough shielding so that you couldn't see it with a radiation detector, but the amount of shielding you would need would exceed the weight limits of most highways in the US. So it's not really a practical thing. On the other hand, plutonium-235 takes about 2 millimeters of lead. It takes about the thickness of your screen on your laptop to shield uranium-235 from detection. And so after 9/11, the detection of uranium-235 became a grand challenge in the nuclear security world. Every seagoing cargo container has enough shielding to keep you from seeing a uranium-235 device, or just uranium-235 on the inside if somebody were trying to smuggle it. There are 48 million of these containers shipped around the world every year, and there is no technology outside of opening them up and hand inspecting the contents that will tell you for sure that you have uranium-235. You can radiograph these containers, but all you'll see is dark objects. You won't know whether that dark object contains special nuclear material, that's the shorthand, SNM, right? Or whether it's just, something that's not on the manifest. If you got this question, how do you determine what that is? Well, if you've got a narrowband 1733 kilovolt laser Compton source, you will excite the primary nuclear resonant fluorescence transition of uranium-235, and you will rapidly be able to tell yes or no that thing contains or doesn't contain uranium-235. That was the origin of most of the funding that we had at Livermore. Now, those transitions are very narrow. They have 10 to the minus 5 to 10 to the minus 6 bandwidth. That means you must have a very narrow source if you're going to make good efficiency with detection. Our analysis suggested if you could get to 10 to the minus 3, remember your 10 to the minus 5 is the natural width, if you can get to something that's within 1000x of that width, then you'll be able to do good detection. If you produce a Compton source, you actually don't produce a monoenergetic source. You make all photons of all energies by conservation of angular momentum and energy. You have all energies coming out, but all the energies are angle correlated. So the beam that I have coming out, if I put an aperture around the beam, I will narrow up the spectrum, and only the on-axis photons, they'll have a narrow bandwidth. And I can keep closing that aperture, and I will make the bandwidth narrower and narrower until I get to a point that closing it just makes the amplitude smaller. The proper design of those sources depends on a whole bunch of physics. It has to do with the electron energy spreads and quality and the laser energy spreads and quality. The net of all of that is if you do things correctly, you can get to 10 to the minus 3 bandwidth out of your Compton source. People who make Compton sources today, who have done science experiments with Compton sources, typically have 10% bandwidth. So this 10 to the minus 3 is a step about 100x beyond where doing things in a naive way would take you. And that was really part of the whole activity at Livermore, was to get to this place with enough photons that you could do detection. If you think about it from a detection standpoint, and I think, I'm going a lot into this nuclear stuff, not just as background, but I think there are places where you can use these nuclear signatures as a different way of making contrast agents for detection of disease. So, bear that in mind when I'm blathering on about nuclear physics. We'll get to the medical stuff in a minute, right? So we have a monoenergetic source. We have a monoenergetic source that has a 10 to the minus 3 bandwidth. It's got a megavolt center energy. So 10 to the minus 3 means it has a keV wide spectrum. I pass it through an object. That object has some resonant material. The light that comes through will have had the stuff that was on resonance absorbed. So I will end up with a notch in the spectrum, right? If I look at that, the depth of that notch is the signal. It tells me how much material was in the way, and everything that's outside of that notch is just the noise that's going on to my detector. So what I really want to do is I want to increase the spectral density, the number of photons I have within the notch relative to the number of photons I have outside of the notch. So I want to increase the signal to the noise. And if I do that, that is the photons per second per EV, because these things are about an EV wide, divided by the actual fractional source bandwidth of the thing that I had coming in. That's what I call the source spectral density. And that's the metric that you use for looking at different applications. If you plot source spectral density versus the inverse fractional bandwidth, you can see a whole range of applications become more and more accessible as you increase the capability of your machine. Down here in the 10 to 100 range, you're doing proof of principle experiments. You can start to do optics experiments at 10 to the 4. You can detect isotopes at 10 to the 5. You can assay materials at 10 to the 6. You can make tomographs of materials at 10 to the 7 or 8. And if you get way out here, you'll actually do fundamental nuclear spectroscopy. The Livermore early machine, the intermediate, the Gen 2 machine, produced a spectral density of about 20. If you go to synchrotrons, there is exactly one synchrotron on the planet that makes a lot of light above 100 kilovolts. That's the European Synchrotron Radiation Facility in Grenoble. And at 500 kilovolts, it has a little over 10 to the 6 spectral density. So I went to the synchrotron in Grenoble. Turns out that the European Nuclear Reactor Research Facility is just across the street, and I know people there. And we proposed an experiment with this synchrotron, and that experiment was to look for lithium-7. Turns out lithium-7 is one of the rare elements or rare isotopes that has a resonance that's below a megavolt, and its resonance is at 478 kilovolts. This one of the beam lines here has a lot of photons at that place, so we went there to test out a particular kind of detector that I had invented called a dual isotope notch observer. And we did a set of experiments. And one of the primary experiments we had was we took the 10 to the minus 3 bandwidth beam. We had a set of, and we put the dyno detector on the other side. And then we had a set of objects that we were going to scan in front of the beam. One was a tungsten block. Another one was lithium-6. It's actually lithium fluoride, but it was all lithium-6 fluoride. And then we had some lithium-7 fluoride, 2 vials of lithium-7 fluoride, some more tungsten, and then a lithium battery. The lithium battery, of course, has lithium inside of it, and lithium is 92% lithium. Natural lithium is 92% lithium-7 and 8% lithium-6. And so we tuned the source to 476 kilovolts, 2 kilovolts below the resonance, and we scanned the objects through the beam. And then we tuned the thing to 478 kilovolts, which is the resonance, and we scanned the objects through the beam. And this is what we saw. Where you have no lithium-7, the signal was exactly the same, right? It's just an attenuation of the object. Where we did have lithium-7, the off-resonance signal, and the difference between the off-resonance and the resonance signal told you how much lithium-7 you had. And in fact, when we had 2 vials, we had roughly 2 times the amount of absorption of lithium-7. And then, of course, when we got to the battery, the difference between the off resonance and the resonance is where all of the lithium is in the battery. And this kind of tells you a little bit about what kind of structure the battery was, whether it was a spiral wind of the lithium or whether it was a pin in the center or whatever. And in principle, you could use this to image. Another application you might have that's based on this nuclear resonant fluorescence is mining. If you do rare earth mining, you dig a lot of rock out of the ground. You take that rock with large trucks to a crusher and you crush the rock into powder. And then you put that powder in a chemical bath and you chemically extract neodymium or samarium or whatever you want to make magnets for your next Tesla out of that solution. And you precipitate out the materials you want, and then you're left with this giant chemical waste remediation problem from the crushed powder effluent that you used. That remediation problem is what drove most of the Western world away from doing mining of rare earth elements because the cost of it was too high. If you had a Compton source that was tuned to the resonances of neodymium or samarium or europium, you could take the rock before you actually crush it, and you could analyze it to see if it actually had the neodymium in it. And if it did, you could kick it off in one direction. And if it didn't, well, you wouldn't crush it. And that would dramatically reduce the amount of rock that you crush. It would dramatically reduce the amount of waste that you have to remediate. And if you go to the mining industry and you ask them about, what impact would this have, their rough estimate is that it would be more than a 500% increase in efficiency of mines. So things that would not be economical to mine would all of a sudden be economical to mine, and to mine in an environmentally friendly way. Okay. So many applications are enabled by this increase in spectral density. ESRF had large spectral density at 500 kilovolts. If you went to 800 kilovolts, the spectral density dropped off the map. It drops faster than exponentially, right? And we can push that very easily in a Compton source to something higher. In fact, an optimized warm Linac Compton source at 2 megavolts could have a spectral density in this range of 10 to the 10, where it enables basically everything that you want to do on this chart. Most of the R&D at Livermore went into solving those problems. If you're going to solve that problem, then you have to make an extremely bright Compton source, because the NRF is very narrow. You have to make it a compact source because the real estate at a port is very expensive. And you have to make it robust because trucks are driving around and it's a busy place. It can't be a pristine lab with vibration isolated tables and everything else. It's a busy, noisy place. And ultimately, the person who's operating the source is not a PhD scientist. It's somebody, that's a dock worker. They have to be able to turn the machine on and go, yes, no, it has something there. If you solve all of these problems, which was what we were doing at Livermore, you can scale that megavolt source back into the 100 kilovolt range. And now you have something that is very compact that can be used to address a wide range of impactful medical and industrial applications. So in 2017, Lumitron Technologies was formed. It was formed based on 13 of my patents from Livermore. And we established the headquarters here in Irvine. Part of the reason why I was the lead hire for the Convergence Optical Sciences Initiative is that the Convergence Optical Sciences Initiative is supposed to have faculty who have connection to commercial activities. And so because I was standing up this activity, I became the target hire for that effort. The company is located at 5201 California. If you don't know where that is in the research park, that's the corner of Bison in California. So it's diagonally across from the med school. Right? It's about as conveniently located as we can make it. The construction of the HyperView KV machines, which is the 100 kilovolt and down machines, has begun. It has the potential for 1,000x higher spatial resolution, game-changing reductions in dose, and ability to elementally assay and image different contents of objects, right? You might ask, well, what does 1,000x mean with respect to increased resolution? This is a conventional chest x-ray. 1,000x, because this is a 1080 screen that I've got here, 1,000x would mean I could zoom into roughly a pixel and zoom it to the full size of the screen, and I would have the same resolution that I can see. Even that's a little hard for you to imagine, so let's do an analogy. This is a gigapixel image of the Golden Gate Bridge. And what we're going to do is we're going to say, this is the 1x resolution image that you have for a chest x-ray. And we're going to zoom in 1,000x and see what you see, okay? So we're going to say this is 1x. You'll notice that this is from a Bentley Motors campaign, and it'll be obvious why in a moment. But let's start with 1x here. If we zoom in 10x, this is what you're going to see. Okay, that's 10x, and obviously there's a Bentley on the bridge, right? Let's keep going. Now we're going to go to 100x. So at 100x, you see that there's a male driver, and that there's you can just start to make out the Bentley logo on the car seat. And at 1,000x, this is what you see. Okay, so now you're able to see the stitching on the seat. So remember, you're standing on the hill looking out at the Golden Gate Bridge, and if somebody's walking on the bridge, you would be able to see the stitching in their clothing at 1,000x. And if you got a tunable monoenergetic source, you would actually be able to tell what the composition of that stitching is. So when you kind of think about it from that perspective, there are a number of ideas that pop into your head with respect to what you might do in medicine. In fact, the last couple of years I was at Livermore, I spent my time thinking about that and writing patents in the medical arena for things that you might do with these sources. The potential for positive societal impact is enormous. I could go on for hours on many of these topics. I'm going to skim through 4 of these on the medical side in the remaining time that I have. The first one is compression-free mammography at 100th the dose. When you do conventional mammography today, the breast is placed between plates and it's compressed. And the idea, of course, is that the woman is going to come in and have that mammogram and then come back at a year or a 2-year interval, and we're going to compare mammograms time to time. Obviously, if you don't compress exactly the same way, those 2 pictures become hard to compare, but there's another problem with this, and that is that this is not a very comfortable procedure for most people. And so you would like to come up with something else that's different. Going back to ESRF, they used a different beamline at ESRF to do a very interesting study. And in that study, they constructed a table. The table had a hole in it, and the breast could hang down freely. And then they could rotate the table slightly in the synchrotron X-ray beam and make 3D tomographs of the breast without compression. Now, if you think about it, if you make this 3D tomograph and there's no compression, the possibility of being able to register one period, 1 year to the next year becomes much higher. And more importantly, well, you can see the cancer in this particular range, but the takeout of this is that it was 1/25th the dose of a regular conventional CT image and about 1/100th the dose of a standard 2-view mammogram at 16 kilovolts. So this was actually, I think, quite a motivational story for going down the path. Another potential application, and actually this is one of the very first synchrotron medical use applications, and one of the things that motivated me, because I was teaching at Stanford when this particular thing happened, is the use of the source to enhance contrast in different imaging studies. So ultra low dose angiography is an interesting thing to look at. When you do coronary angiography, you will inject into the bloodstream a contrast agent. It's generally an iodine-based contrast agent. The blood vessels, of course, don't have much contrast for x-rays that could penetrate the full body. The iodine adds the contrast, and you have an ability to see the blood vessels. The problem with this is the amount of iodine that you administer is sometimes toxic to the patient. People do die from the levels of contrast that are administered, and it requires exposures for one image that are at the annual limit for one sitting. So you don't really do this unless the person's pretty sick. And what they did at Stanford was they took the small synchrotron at Stanford and they made it so that there were two beams of x-rays coming out of that synchrotron, one of which was tuned just below the K-edge of iodine. The K-edge is the energy at which you remove the 1s electron from an atom. And every atom has a different K-edge, so it has a different X-ray signature. And the cross-section for absorption of X-rays changes by nominally an order of magnitude as you go across that edge. So if you take an image above the K-edge and an image below the K-edge and you subtract them, really all you're seeing is the thing that has to do with the edge. So this is a great way to increase enhancement. And so this is a 33 above, 32 below, and then the subtraction image, getting rid of a lot of the other objects. This wasn't actually the Stanford data. This was data taken on a pig heart many years later. Okay. Another application, detection and treatment without patient movement, all at the same time. Okay, so we're going to start with this cartoon model of cancer attached to a blood vessel. The dark objects are meant to be the cancer cells that are growing. The purple is meant to be a drug that has been specifically tailored to latch onto the proteins that are on the outside of the cancer cells and go to the cancer cells specifically to them and nothing else. One strategy of trying to treat cancer in this way is to attach to those cancer-seeking drugs some radioactive element. There's an entire industry of people who do this. In fact, there were people at Livermore who were doing americium chemistry to try and do this kind of path. There are a couple of clinically relevant problems with this. One is that the compounds that you give to the patient also accumulate other places in the body, like the thyroid and the lymph nodes. So you might cure cancer in one place and give cancer somewhere else. And then there's another practical problem, and that is, let's say it only goes to the prostate and you're trying to cure prostate cancer and you've cured the cancer. As long as the compound is still in the body, you're still giving radiation to the body. And so you might cure it, get to a healthy state, and then if the body hasn't flushed it out, you're causing damage again. In the Compton approach, we can do something slightly different. What we do is we attach to that same molecule a benign atom, gadolinium, gold, platinum, something that's high Z, low down in the periodic table. We do high Z because the K edge of high Z is at high energy. So gold is around 80 kilovolts. You're transparent at 80 kilovolts. So there's lower dose to you at 80 kilovolts. And we use the K edge differential absorption to locate where the disease is. Once we have located the disease, we can then turn up the X-ray source at above the K edge. Now, when you remove the electron, the K edge electron from an atom, you leave the atom in a very excited ionic state. And it relaxes by having a 2p electron fall in to fill that 1s vacancy. A 2p electron flies off to conserve energy and momentum. A 3d electron flies in to fill the 2p vacancy. A 3d electron flies off to conserve energy and momentum. And you get this cascade of ionization called an Auger cascade. Those electrons that come off of that K-edge ionization event don't have enough energy to travel very far. They deposit their energy locally to where the gold or platinum or gadolinium was, and locally is where the cancer is. So now you have a way of actually seeing the cancer with very high spatial resolution and giving dose to the cancer specifically with very high spatial resolution. So you would have a conventional X-ray image, a subtraction image might show you the breast problem, and then we would go in and we would try and treat that region directly. In principle, you could do all of this without ever moving the patient. You have the patient in front of the machine, you see the image, and before the patient moves out, you do the treatment. Phil Stricker is a world expert in robotic surgery. The urologist that I quoted at the beginning of the activity. And his comment was, we can find the disease with different diagnostics, but then he has to put an X on you with a marker, haul you off into the operating room, and you're not a solid object, and everything moves around. And then he's left with two choices: take too little, which isn't good, or take too much, which isn't good, right? So this is really a different route of doing things. The last one that I would tell you about is in vivo small animal radiography that enables rapid drug development. When you do a drug trial, and especially as you're going along the path to human use, you start with mice, and you don't image an individual animal through its entire process of the treatment. What you do is you create clones of that mouse. You don't image the mouse because the mouse is very small. It's 100 times smaller than you, and you don't have the spatial resolution with a Bremsstrahlung tube to do that. Typical Bremsstrahlung radiation for a 60 kilovolt chest X-ray is around 500 to 600 microns, right? So it's just not the resolution. So you clone the mice, you give the disease identically to the clones, you give the treatment identically to the clones, and then you kill them off one at a time, and you open them up, and you follow what the progression of the disease is. If you've got a very bright source, in this case, I'm talking about the synchrotron in Japan, you can image the mouse with much higher spatial resolution. This is actually a 3D tomograph of a laboratory mouse. You can see the whiskers in this picture if you've got the resolution on your screen. I actually have versions of this where you can zoom in on the ear and you can see the 10 micron bones in the ear in the mouse. It is astounding, right? The resolution that you can have. Here's another one. This is a mouse embryo. This guy is 2 millimeters tall. Because the radiation comes from a very small source, you can actually have phase contrast imaging that isn't based on the density of the electrons, but it's based on the phase difference between different organs. And so this tomograph, you're actually going sectioning through the object, through the animal, and you can see all of the organs, the bone structure, everything in the embryo in that picture. That picture took about 10 to the 11 photons to make. The machines that we're trying to create right now are supposed to make 10 to the 12 photons per second, right? So that kind of gives you the scale of what we're shooting for in this setup. If you talk to pharma, it costs $1 million a day to bring a drug to market. It's pretty easy to see that there might be an advantage here to doing this, where you take the 10 mice and give them the disease identically and give them 10 different treatments and follow each one of the mice individually to see what happens, right? The construction of a 4th generation machine is on the way over in 5201 California Avenue. We actually are vertically integrated in fabricating accelerators and lasers in that building. If you want a tour, I'm happy to give you a tour of what we're doing there. We're making our own parts and have established all of the fabrication technologies. We've also established a rather large, almost $12 million contract with DARPA to create a 10 kilovolt to 100 megavolt to 1 megavolt capability. This is part of their gamma ray inspection technologies program. We have a project called Determination. So we have GRIT and Determination as the two project acronyms. The project itself has specifications that are in line with what we're doing, but it also has a size, weight, and power requirement that the whole machine at a megavolt has to fit in a seagoing transportainer. It has to have a weight that you can put on a C-130, and it has to consume a power that's no bigger than a military diesel generator. So there are extra gotchas in doing that activity. This activity will step systematically from 100 kilovolts to 300 kilovolts to a megavolt over the course of the next 9 months. And we're working on that over at Lumitron. To make a megavolt machine, we have to make 150 megavolt class electrons. And if we don't have a way of decelerating those electrons, which we are not doing just yet, you need to dump those and you need to deal with the radiation that's created. And so we have created at Lumitron an enclosure for that initial system. That enclosure is a $3 million project in its own right. We started with the other half of the building. One day later, we had all of the offices out of the way. A couple of days later, they've got all the ceiling out of the way. They started dicing up the floor. The floor in the research park are about 5 inches thick. They can't handle the weight of the enclosure. So we had to excavate, pour our own concrete for the enclosure. There's the rebar for the enclosure, pour the own concrete, and then build up the structure outside and start filling it with lead bricks and borated polyethylene. As we move forward, and there's the completed enclosure. This enclosure has 600,000 pounds of lead. Each one of these doors weighs 25,000 pounds. Each one of the doors weighs 25,000 pounds, and the hinges are so good that you can actually pull them open and close them. It takes a little bit of leaning, but you can still do that. And you can see we've started to install the accelerator tables every day. This thing is changing. This was about, I don't know, week and a half ago of where we were. We did the ribbon cutting for the overall enclosure last Friday, Friday the 5th, for the machine. The nominal 100 kilovolt machine will come online in the May timeframe. This is the cutaway of the 100 kilovolt machine. The sections for the megavolt operation have been removed or not going to be in place for the 100 kilovolt arrangement. Laser sits outside the table. The RF power sources are here, and the X-rays come out this hole in the door, right? And all the X-ray experiments sit outside here, or they sit inside the bunker, and the detectors sit outside of the arrangement. One of the primary initial things that we want to do with this setup is look at a set of mouse models using gadolinium as a contrast agent and potentially using the electron beams to do flash radiography. For those of you who know Charles Lamolli, Charles is a big proponent of flash radiography. Flash radiography is the rapid deposition of dose in the patient. You give the same fraction to the patient, but instead of giving it over an hour or over several days, you try and do it in a microsecond. And while the mechanisms around that are still there's a bunch of debate, the efficacy is not in debate. You do more damage to the cancerous material and less damage to the healthy tissue in the flash radiography protocol than you do in the conventional protocol. There have been a number of studies that have showed that, and we're going to try and do that with the Compton source, with the idea being that we could image the disease and treat the disease in place. This whole activity around using Compton sources and the various different diagnostics, theranostics, treatment modalities that it enables are part of the 2021 Chow Family Comprehensive Cancer Center renewal grant that was submitted in the end of January. And so with that, I'm going to stop and say the required facilities and funding and staff are now in place. We're going to be doing some of this activity over in the building here. Hatham and Trevor have our work in my lab that is in the Beal Applied Innovation Center, but they will be part of the experiments that go on with the first light of the machine that is coming online this year.
Well, thank you so much, Dr. Barty. That was an awesome presentation. Let me give you a virtual round of applause. Thank you, thank you. If there are any questions, feel free to raise your hands, or honestly, if you want to just shout out, I think we can try raising hands and I can call out. We don't have too much time for questions, so if there are any questions, please feel free to ask now. And if not, I can warm up the waters a little bit with a quick question. Kevin, sorry. Oh yeah, I meant raise. Yeah, good. I didn't see your hand. Go ahead.
My question was actually about the cancer application using, I guess, an antibody tagged with the gold bead that would be excited by the laser. So one of the most successful immunotherapies right now is the CD19 like CAR T cell, basically antibodies targeting a type of cell which is in the blood. So I'm wondering, so if you, for example, you use this antibody, it's gold plated, and you're targeting a cell that's in the blood, so within all of the vasculature. I'm just wondering about the electron range. Am I about to fry all of my patient's vessels? If I,
So I think this is a very good question. This technique of Auger therapy, if you can make it so that it really works the way that we talk about, it can be used on distributed cancers, like the ones you're talking about. When you're at 78 kilovolts, a chest X-ray is a 80 kVp peak on the tube, which has a peak X-ray distribution around 60 kilovolts. And it's really the 60 kilovolt photons that are making the shadowgraph. The higher you go, the less absorption there is in the body, right? Except, of course, where you've got the gold or the platinum or whatever else that's big and heavy. And so you could imagine, illuminating a wide area, giving a relatively low dose to the healthy tissue because it's very transparent, but still giving a high dose to the cells that you care about, the ones that are attached, that have the contrast agent attached to them, right? So yeah, I think this is a clear thing that we're interested in doing. I think there's a bunch of chemistry that also needs to be investigated around the generation of electrons. That using those electrons in more than just a heat source to affect modifications.
I see. Yeah, because I'm thinking, like, for example, you have a cell in the blood, it has this, gold bead, but it's only, it's pretty close to a blood vessel. Say it's going through a capillary or something. But yeah, I guess another, if you could use something beyond a heat source, for example, to like cause like apoptosis or something like splice mitochondria or something like, yeah, okay, totally. Yeah, awesome. Thank you for such a fascinating talk. And
you're welcome.
And congratulations.
Hi, Dr. Barty. Wonderful talk. I'd like to ask a question as well. For your like tomographic reconstructions, what is your scanning geometry? Since it is such a narrow beam, are you doing like a parallel geometry? And then with that, what's the likelihood of translation into, for example, like a rotating CT system?
So you're not going to do CT in the method that CT is done now. You're going to take a different modality for CT. Because you're not going to make a rotating source around the patient that's a Compton source. There are methods for modifying the output beam profile from being a pencil beam to being a fan beam to being a cone beam that are all monoenergetic. And there are also technologies for doing a scan of a pencil beam, right?
Yeah.
So, you could imagine raster scanning an object.
Yeah.
Generally speaking, when we think about the applications in humans, we limit ourselves to being able to translate the human in one dimension, right? They can move on a table in a linear fashion, but we can't spin the human and we can't raster the human in 2D. So we have to do something with the beam that allows us to do what you're talking about. And so those are some of the ideas that are out there. But in the CT world, because you've got a monoenergetic source, you likely won't need as many views to get the same information. Part of the problem with a CT machine is that the light comes out in a cone and it comes out polychromatic. And so determining what actually caused the attenuation on the other side of the object is difficult without a lot of views, right? You don't know whether it was attenuated because you went a long path or whether it was because there was a lot of low energy photons that were absorbed or high energy photons that were absorbed. All of that kind of ambiguity goes away. In fact, one of the more mundane, but I think very important applications is that making a very collimated beam with this would allow you to do very precise bone density measurements. So if you're looking at osteoporosis and the change in bone density, That's also a hard thing to do with a conventional X-ray tube, because again, you've got polychromatic light coming out and it's going out at an angle. And so if you're trying to do a Beer's law absorption in a linear path, you don't really have a clear way of doing that. But if you got a monoenergetic source that's really a pencil beam, then you have the potential for doing those measurements much more effectively. On the other non-medical side of things, I think the first application that we will be pursuing is certification of additively manufactured parts. If you think about additive manufacturing, 3D printing, when you 3D print something, you print at a scale that's beyond the resolution of conventional X-ray tube, and you don't really have a way of certifying the part without destructively dismantling it. This would allow you to certify the part, and those things you can move in 3D in the beam. So, your tomography is a much simpler issue. Now, we have been talking with people about, artificial hips and things like that that might be 3D printed, and whether you need that same certification process. Right now, there is no certification process that is universally accepted for 3D printing.
Well, thank you so much for the questions, everyone. Unfortunately, I'm going to have to end the conversation as we transition into our student presentation. So everyone else, let me thank Dr. Barty again for the talk, and thank you for the questions. Thank you again for coming out to our retreat, or our virtual conference, I should say.